everyone. Welcome to our next season of Mycological Society. I'm David Bermudes. I'm the president of LAMS. And before we go any further, I'd like to ask the council members if we have any announcements.
Say that, hi, I'm Jess. I'm the newsletter editor, and I'm always open to you guys having photos, what you're finding out there, any little stories of, if you're just out in the woods and finding something interesting, write a little story about it. I think it's fun to have in the newsletter. Any photos, whatever. We can have some cool articles. Always open for submissions.
Anyone else? Okay, so I have the pleasure of introducing myself, which I already did. I'm a professor at California State University, Northridge, and I mostly do research on bacteria, things like salmonella, and things about like how they can kill cancer cells. But that's not what I'm talking about today. I'm talking about my other passion, which are the fungi. And I won't be talking about my research, but I'll be talking about this explosion in information that's come through DNA sequencing that's told us so much about the early origin and evolution of life on Earth. So I'm just going to take you a walk through my own trip, so to speak, and we can see what we think about who the first fungi were and what they share in common with animals. Now, I didn't realize it when I said yes to speaking today, but this is actually my third presentation today of different things. And I'm losing my voice a little bit, and I'm just bear with me. Well, one thing I want to say is I do have a passion for fungi, and I do some research with them a little bit. And I'm in an old building at Cal State Northridge, and I don't even have to work to find beautiful and interesting molds. They come to me. We have a core facility that has all the Petri plates for all the microplastids, and everyone else is finding these plates and throwing them away, and I'm going in there and I'm taking them out and looking at them. So I don't have anything specific to say about these, but here's another one before I get on with what I'm going to talk about tonight. This was a random isolate in a Petri dish, and it had this weird yellow goo coming out of it, oozing out of it. And you can see I took a scalpel and I cut a little piece out. And a couple days later, the ooze was growing on its own. And the ooze turned out to be bacteria. In fact, it turned out to be 4 different strains of bacteria. So this thing that I'm interested in on the side is that this fungus just happened to land on this plate. There's no other fungus. The plate would be, like this. There's no other fungus anywhere else. There's no other bacteria anywhere else. So my hypothesis is that they got there together, that they traveled, that the bacteria are traveling on the spores. And there's more than one of them. So pretty interesting. In fact, just before COVID hit, we had to stop this. But I had a high school student from Granada Hills, and she was growing these bacteria. You can separate them with antibiotics and reintroduce them. And the bacteria swim underneath the hyphae, using the hyphae like little highways. We actually made a little movie of that, just swimming back and forth along them. So I hope to get back to that someday. So this is what we're going to talk about today. You could call it the origin and evolution of fungi. We're going back very far, way past the dinosaurs, way past the Devonian and the early plants, all the way back here to, really not long after the origin of life on Earth. So I want to remind you that our planet has changed over time. It's evolved. it started as a much smaller aggregation and was bombarded from space with things like what do you call them, comets that carry a lot of water. It's probably where a lot of our water came from. It's where a lot of the mass of the Earth came from, in addition to the initial accretion. And it's changing with time, right? It's the early Earth is anoxic, no oxygen that we could breathe and survive on. And of course, the continents were in different locations at that time. They changed through time. They're different now than they were when the dinosaurs were here, and much different way back then. And then you'll also notice that there's some organisms drawn on here. And this is meant to represent a kind of timeline going, it's in German, I hope you read German. So, but you know that a lot of these cognates you can recognize. Some of these are pretty easy. So 4 MRD is going to be 4 gigaania, or 4 billion years. 3.8, that's when we think life started. Archaea in German is archaea in English, because that's a group of bacteria. Proto-eukarya is pretty close to the English, and that's what we're going to talk about first, is the origin of eukaryotic cells that go on to give rise to the fungi. And in fact, you might know the word pilts, like steinpilz. There's the word pilts there for the fungi, the protists, and the plants. And this diagram goes off with the animals down here that I haven't shown. So this diagram is showing you some important things that happen to the Earth's atmosphere. And I'm just going to talk about 2 of them, which is oxygen and carbon dioxide. So early Earth is mostly carbon dioxide. You've heard about the rise in carbon dioxide in our atmosphere, and in my lifetime it's gone from maybe 0.034 to almost 4 now. But in early Earth, the atmosphere was 95% CO2. If you look at, so the other one to look at is the oxygen line. And you'll notice 3 little humps there. This early one, debated too, by the way, almost everything I talk about today can be debated in one way or another. But there seems to have been some chemical oxygen present off and on. But the organisms living during this time are essentially all anaerobes. And a lot of them, early on, are what are called chemotrophs. They're actually living off chemical energy, like you see in the deep sea hydrothermal vents. Sometime in here, maybe, is the first oxygenic photosynthesis with cyanobacteria, fixing carbon, releasing oxygen. And that maybe accounts for this little bump. Then we have another little event that might have been the origin of algae, eukaryotic cells, that started making bigger structures, inhabiting more area. And then finally, the advent of the vascular plants that colonized land and increased oxygen significantly. No, I don't need that. So this is a kind of evolutionary tree I think everyone here is familiar with, because we look at related traits like a vertebrae, and intuitively we all know these subgroups like birds and fish, and yet we also know that they all belong together because they share common features. So we're going to take a look at fungi like that today, but we won't have the benefit of large bony structures like vertebrae. We're going to have to look, starting with the mushrooms, but then moving backwards into just cell biology and the machinery that works inside of the cell. So you can answer the question, what was the last common ancestor, pretty easily, just off the top of your head, which is it was an early evolved eukaryote. But what does that really mean? Well, first of all, prokaryotes like the bacteria, some of the archaea we're going to see later are much different than this. but they're pretty simple. They don't have complex inside. They just have a nuclear material not bounded by a nucleus, compared to a eukaryote that has a nuclear membrane. The eukaryote also has this elaborate Golgi ER system that's used for protein synthesis and protein secretion. Trafficking of vesicles of different enzymes that are delivered to different places is highly complex. Most of that is absent. Most bacteria have a cell wall, simple cytoplasm with ribosomes, no nucleus, no Golgi ER, no significant vesicular trafficking. They have this very simple structure that's referred to as a flagella, but it's not like your flagella at all, not even related. So some of these things, and here's a paper where they put it on a tree. So Leca would be last eukaryotic common ancestor. Feca would be the first eukaryotic common ancestor. And here's some of the things that are thought to happen in between. And I've just listed those here too, as things that we're going to look for in the past and look at how they relate to the fungi. So the endosymbiotic theory posits that eukaryotic cells evolved from symbioses with prokaryotes, and that really eukaryotic cells, meaning all of your cells, are chimeras of a eukaryotic nucleocytoplasm with a mitochondria that's not related to your nucleocytoplasm at all. You have very ancient members of the prokaryotes living inside your cells as mitochondria. Plants have them in their cells as plastids. And you can determine that. This was finally proven just by sequencing. So, first of all, it was known for a long time that mitochondria and plastids had features that were very similar to free-living bacteria, or bacteria that are symbiotic inside of cells. If you look at them through the microscope, they divide. They don't come from nothing and disappear. One always makes another. The kind of division that they undergo is similar. It turns out they have their own DNA. They have their own genome. And their genome has their own ribosomes in it. And the ribosomes make the protein, some of the proteins that are made inside of the plastids and mitochondria. This is thought to be the remnants of ancient DNA that is in here and in here. In fact, when you track it, your eukaryotic DNA, your nuclear DNA, is going to track to a distant archaeal lineage that we'll talk about a little bit more. Your mitochondria are closer to nitrogen-fixing bacteria, the alpha proteobacteria. And perhaps not surprisingly, now that you get where we're going, chloroplasts are actually very closely related to free-living cyanobacteria. So here's the Logi archaeum. And what I want to point out is on that slide where we talk about all the differences between prokaryotes and eukaryotes, that's mostly really the bacterial lineage versus the eukaryotic lineage. Turns out to be much less of a chasm when you look at some of the archaea. And the Logi archaea bacteria, they have some of these things that I was saying don't exist in typical prokaryotes. So they have cell division cytoskeleton. They have endosomal sorting, something like Golgi ER, and that trafficking machinery, the ability to deliver vesicles precisely within inside of a cell. So this is, in fact, actually where your nucleus traces to. It gets its name from Loki's castle, which is a deep-sea hydrothermal vent off of Norway. So that's where your nucleus is probably from. So another thing to say about eukaryotes is this is a cross-section of a cilium. In eukaryotes, a cilium and a flagellum are the same thing in terms of the ultrastructure. Flagellum is just when you have one or two. Cilium is when you have many, but they're the same structure. And you can tell that by electron microscopy because it gives what they refer to as a 9+2 microtubule organization. So these are doublets of microtubules, and there's 9 of them and 2 in the center. And that's the flagellar shaft that you see if you've seen that under the microscope. Underneath that is the centriole, and also referred to as microtubule organizing center. And its ultrastructure is a set of triplets. This structure is fundamental to cell division. When a cell like this is going to divide, which is what you see in this slide, it actually has to shed this structure. This structure detaches from the membrane. It actually divides on its own. Then it migrates to the poles and organizes the microtubules that are stained in green here. They're stained in red, and the chromosomes are stained in blue. So this motility and cell division is really an ancient and fundamental part of eukaryotic biology. So timeline, where on that first slide might we be starting? Well, one of the things, actually you may know this, we have cholesterol. Fungi don't have cholesterol, they have a variant referred to as ergosterol. Sterols are fundamentally a eukaryotic molecule, and there's actually a fossil record of them. Not the ergosterol and cholesterol, but the precursors, which are sterins. And those have actually been found all the way back to 2.7 billion years ago. So that's a possible timeline for the origin of eukaryotic cells. In the geologic record, we're talking about a very imperfect record sometimes, incomplete preservation, not widespread distribution, but these are some of the most best supported ideas right now. And so you see it's actually also giving a couple of dates here. I'm going to try and line that up with our other timeline, which was the Earth's development. And so some of these things might line up, or maybe it's just coincidence, but let's look at them. Okay, mitochondria. They're often, I hear, powerhouse of the cell. They're aerobic. They need air. They respire air. They produce ATP. So if you're going to have a symbiosis based on mitochondria that's aerobic, you need air. So it needs to have happened after the origin of the cyanobacteria. And we probably need to have a little bump in the oxygen. And so, in fact, I don't know if they were looking at the same thing, materials at the same time, but that merger that creates that symbiosis that creates the mitochondria somewhat correlates with the time when there starts to be air that would support that kind of association. But later on, I'm going to throw you a little curveball when I talk about the chytrids, because the chytrids might have a little secret for us about another possible metabolic origin of the mitochondria. So here's a simplified, phylogenetic tree. Of course, the way we're reading these, and I should have pointed it out sooner, is what we do is we trace back to a node. The closest node tells you the closest ancestor. So ascos and basidios are the closest among the fungi. Then you go back to this node for glomerulus, back here for zygos, back here for chytrids. Some people, including my PhD advisor, didn't think chytrids were really fungi, and I'll explain why she thought that. And this is sort of really my point of departure for this talk. The other thing I'm going to point out is, this is maybe 10 years or so ago, it's mentioning paraphyletic, and I'll show you what they mean by that. When you do a finer, unrooted tree, well, here's the chytrids. They're two different lobes that aren't really touching each other. They have more common ancestors with the zygotes. And you'll notice the zygotes have some very deep branching too. So they are groups among themselves. That's what they're referring to as polyphyletic. And that's the kind of thing that people are discovering with more and more DNA sequencing. So, I had this argument with Gary Linkoff. And I only met Gary maybe 2013, 2014. You see him there with Florence. Florence and other members of LAMS knew him all the way back to the 80s, maybe even late 70s. And so we were at the ARB for the fair, and I think we were in the kitchen, and we started talking. And somehow it came to chytrids, and he said, well, you know, do you really think they're fungi? And I said, yes, I do. I said, well, you know, I've had this argument before. I had it with my thesis advisor, who you'll see in a second, is Lynn Margulis. And he said, I had that argument with her too. So we had a little three-way thing going. And I'll show you why I agree with Gary. And Lynn's is to me a more sort of artificial construct, but I'll show you what she was thinking and why she was thinking. It's not that it doesn't make sense, and that's why a couple slides ago I said, well, it's a matter of semantics, it's how you put it. If I say here's an Amanita muscaria, and it's big and spongy and complex, and it has complex division, and it goes through this life cycle, that's what a fungus is. Well, now you're kind of stuck, right, to describe maybe something like a chytrid that's not so much like that. And I actually brought a couple of chytrids to pass around. They're really interesting. They don't grow quite like other fungi. Really? Okay. All right, well, save them for me next time. Yeah. So that's what I mean by a matter of semantics or perspective. And also, a lot of that's being changed by DNA sequencing. So yeah, I had that argument with Gary and then later with Lynn. So with Gary, it was, yes, they're fungi. Why are they fungi? Well, I'll show you some more slides in a minute. But, you know, they have chitinous filamentous threads, which is the fundamental thing about a fungus. They're not exactly the same. I'll show you what the differences are. But that's pretty fungus-like to me. You make spores. They have both haploid and diploid phases. So it was, and they're osmotrophic, which is one of the essential things you think about with fungi. But for Lynn said, well, all the fungi I recognize, Ascospicidium, they're non-motile. That's part of what I see as a fungus. See, what Lynn was doing, this is a modified Whitaker scheme, the 5 kingdoms scheme. And for Lynn, the ones up at the top, the plants, animals, and fungi, are all complex multicellular organisms. And of course, plants and animals really have embryology on top of that. A very, complex initial development stage. Fungi, less, but still fairly complicated. And that's why she said, yes, they're up there. She actually took algae out of the plants and put them back in the protists. And there, the chytrids sat for her as well. So we can start with that idea. What is a fungus? What do we recognize as a fungus? What are the attributes? We can start with the Eukarya and go backwards. I don't know if I need to go through all of these things with you. I think this is the group that most of the people familiar with mushrooms are familiar with. But they have chitin. We're going to talk about that. And of course, chitin is something you find in animals. All the crustaceans, shrimp, etc., have chitinous exoskeletons. They have hyphae with septae. So that's a little bit more complicated than some of the earlier evolved fungi. They also have anastomosis, which was drawn by Buller here, these little cross sections there where hyphae actually fuse with other hyphae and make a network. Seems like a fundamental feature. There might be a fossil record of that, in fact. Fungi, they basically live in their food, no matter what kind of trophic form they have. They're right there. They're not seeking it out. They're either breaking something down, they're symbiotic with tree roots, for example, or they're necrotrophs, being parasites. But they're using this method. They don't have phagocytosis, although they do have pinocytosis, and I'll mention that. So their fundamental nature is osmotrophs of a filamentous network. Also just want to mention, you may not be as aware, but they have this complicated hyphal tip. So the hyphal tips are the parts out there. That's the part that grows. That's the part that secretes all the enzymes. That's the part they grow into after they secreted the enzymes and absorb the products. And they have this complex structure referred to as a Spitzenkorper. It's a microtubule and actin organizing center. If you're going to grow at the apical tip, all the enzymes and chitin that you need has to be mobilized to the tip. So if you look right here, the fuzzy layer, that's the chitin and the beta-glucans. So some of these vesicles here are chitinosomes that have to fuse with this membrane and be released to become part of the growing tip that needs new chitin. It also has this dense collection of membranes, and that's all of them, converging on that one spot. And then I've also shown actin. That's the other cytoskeletal protein. Actin is also involved in the tips, and you also see these little points right there, and those are the points of pinocytosis. So that's referred to as cell drinking, where they can actually take in some of the liquid directly. And that becomes part of my argument for what a chytrid is or isn't. Now this is the other part. This is the part, I don't know, somehow I love this feature, which is they're dikaryotic. You're mostly diploid, and you are only dikaryotic for maybe an hour or two in your life when the sperm first fused with the egg and the nuclei hadn't fused yet. That's your entire history of being dikaryotic. But ascos and basidios, they spend most of their life. The thing that makes their mushrooms is the mycelium underground, and it spends all that time as dikaryotic. If you look closely, the fungi stay paired. So after they make that initial pairing, when the nuclei divide, the pairs stay together. So it's like not only are the fungi mated together, but the nuclei are mated and bonded together. We're not going to go through these life cycles. I'm just reminding you. Here's the phases where we're haploid dikaryotic, N plus N. Here it is in a mushroom. So the mycelium, the mushroom, even all the way up to the basidia, it's only in the basidia that those nuclei finally fuse and then undergo meiosis. So when you think of the humongous fungus, you have whatever 100 square miles of haploid dikaryotic living for maybe thousands of years that way. So here's a chytrid. So chytrids are chitinous. They have these rhizoids, and then this bulbous part that produces the zoospores. This part is osmotrophic. It does not generally have nuclei, though. In a fungal mycelium, all the hyphae are populated with nuclei. In a chytrid, most of the rhizoids are not populated by nuclei, so there's a little bit of difference there. They spend, just like you, very little time as N plus N. They're flagellated, so that's why Lynn didn't like them, or didn't think they belonged as a group. But of course, that does not deter me. They also have a diploid thallus. They don't really spend a lot of time N plus N. Chytrids do some weird stuff, though. Inside of their cell, some chytrids, there are three main groups of chytrids. I showed that polyphyly. This group, the Neocala mastigomycota, lives inside of cows. And I think you all know that cows can't digest grass, but chytrids can. So that's how they're able to, be a larger symbiotic unit together. But chytrids this particular group doesn't have mitochondria anymore. It has these things called hydrogenosomes. And they have a different pathway. It's not the one, if you learned the one for mitochondria, it's not that one. You don't have to learn that one tonight. You just need to know it's different and that it's fundamentally different. So, in addition to eukaryogenesis occurring based on, aerobic respiration, another hypothesis is that maybe early mitochondria had this hydrogen metabolism. Maybe that was part of the early basis for the symbiosis. So here's a look at a comparison someone made of the different features. So rhizoids versus rhizomorphs, septate hyphae. I forgot to mention it, but the chytrids are generally non-septate, but they do show a few here and there. They do make some walls. And so there is kind of a difference between these fungi. You can see how the features taper off as you go back. And what we're going to do now is go back to a couple of these other groups and see how they look compared to the fungi and see how they look compared to animals. Another thing about the rhizoids of a chytrid is that here's an example of actin staining. And I was hoping to find, this is not my work, I was hoping to find this Spitzen work, but I couldn't find a picture of one for chytrid. But it did look punctate as if that were pinocytosis. And the purpose of this paper showed that the rhizoid really develops a multicellular fungal mycelium because it grows larger or smaller in response to food availability. So all of this sequencing has generated information that's told us stuff that we weren't sure was related to this Opisthokonta group, this group with hind flagella. But that's what sequences have revealed: microsporidians, cryptomycota, phylida, and nuclearidida. And I'm going to talk about three of these, and I think we can use that to come to some kind of picture about what early fungi and the common ancestor with animals was. So here's the microsporidians, and these were really enigmatic for a long time. And some people thought they might be related to fungi, but it really turns out they are related to fungi. They do have chitin. They do have a mitochondria. They have mitosis and meiosis. So that means they're both haploid and diploid. They don't have flagella. And we'll see a tree later on that talks about the fact that some of the groups of fungi have retained flagella, like the chytrids. Others, like the ascobasidios and zygomycetes, all lack the flagella. And we'll see here, for example, yes, it happened again for the microsporidians. If you don't know, we don't need to talk about this tonight, but they have this bizarre structure, which is a polar tube. What it is it's a coiled filament. So this is just a slice across it. We only see edges. What it does is, this is a parasite. It fires that out when it comes in contact with the cell that's going to parasitize. It actually translocates its nucleus into the cell. takes it over and makes new microsporidians. It also has the smallest genome of any eukaryote. So the aphelids. Well, these are parasites of phytoplankton, and there's so few of them, and they were so poorly known. But there's a feature here I want to point out to you that we've said doesn't exist in fungi, and that's phagocytosis. They have a phagocytic stage, an amoeboid stage. So this idea that fungi are non-phagocytic, when you go back this deep in the lineage, you can find members that have phagocytosis. And you actually see these, they give them a weird name here, but this is a little phylopod, an extension of the cytoplasm that has the phagocytic ability in addition to this amoeboid stage. And then finally, the Nuclearidida, if I'm saying that right, turns out to be an Opisthokonta amoeba. And that's also only appreciated relatively recently. So it's deeply rooted in the holomycota. By holomycota, that's also a perspective. That's everything we know that's closely related to the fungi, right? Holozoa is everything that's close to what we recognize as animals. And so, indeed, they're amoeboid. They don't have chitin, but they're amoeboid. They have mitochondria, they're phagocytic, they're mitotic, and they also have the genes for meiosis, though no one's observed them doing that. So they're diploid. But it's this pseudopods that are so suggestive of how you might have gone from an amoeba to a filamentous organism. So this is a phagotroph, right? It's engulfing food. Earlier we said the fungi we recognize, most of them are osmotrophs. So somewhere in there set up the potential to make that kind of switch from phagotrophy to osmotrophy. And they do some weird things too, really weird and beautiful. They secrete silica and then they glob it onto themselves like this Bermudes layer. So perhaps even suggestive that they needed protection of something like that, chitinous exoskeleton. No, I meant to leave that one out. I'm just going to skip that. One other feature I want to look at that was looked at in this paper is one that's not quite as certain, but chitin synthase, right? That's the thing that's going to make the chitin of the animals, it's going to make it for fungi. Well, it turns out there's 4 of them, and they're not that closely related. And this diagram color codes where you find the different 4. And so it's the red one that's primarily the fungus one. This is no longer Opisthokonta down here. So there's our Opisthokonta, referring to hind pole, single polar flagella. And so you mostly find the red one here. It is found over here. I don't know. Seems like maybe that could have been lateral transfer. I'm less certain about that part of it. They showed this kind of arrangement to metazoan class and then fungus class. It's mostly not. So I don't think we have a good answer to the origin of the chitin synthases. That's my feeling. Well, this was a book I went through recently, and first 3 billion years of life on Earth by Andy Knoll. And what he did was he went through a phylogenetic tree and added a bunch of fossil pictures and put them on the tree. So here's a testate amoeba. Here's one from the Grand Canyon 750 million years ago. And here's something that looks almost exactly like it today. So pretty good fossil evidence that amoeba had diverged at least by 750 million years ago. But he forgot to put any on the fungal tree, so I thought that's what we could do. So I think many of you have seen the pictures of the fungi and the true mushrooms in the embryo. They had an issue of it with fungi magazines. Probably had it a couple times. And I don't know, is that a marasmius, or it's almost a genus you could recognize? I don't know if anyone wants to guess. Yeah, possibly. Yeah, it really does. So that's 99. Well, that's recent, right? In this scale of thing. Right? The dinosaurs are still alive. There's just a little bit older one. I like it because it just looks like a mature modern mushroom from Brazil, from when Gondwana land was still all together. That's how it got its name. Go back a little more. So now there's a bunch of ones that occur 400 million years ago. So now you're all the way back in the Devonian. And that's early land colonization, maybe not. I'll show you some other examples in a minute. But here's what's referred to as a folioslypid, these leaf-like ones. And these fossil remains look remarkably like that. And of course, that's a symbiosis in itself. Usually green alga with an ascomycete, and sometimes it's a basidiomycete, a fungus, and sometimes it's a cyanobacterium, a photobiont. But that allows colonization of land. It's also good at breaking down rock and making soils. So it's certainly contributing to the colonization of land. But we're going to go back even further. Well, here's just some more, also around that time. So that's the blastocladium mycota. And that's remarkably good preservation there. So not too much of an argument there. Here's another one that I find from time to time. Apparently this is found relatively frequently, but not in high abundance. Thought to be some kind of saprophyte. I think a lot of you have heard this one. And I included it today because I knew someone would ask if I didn't include it. Really hard to imagine why this thing would need to be there and be so big at that time when it had so few competitors. There was a recent paper from just last year, and it got some good preservation and did some sectioning. It was able to find that indeed there are tubules in there, so that's certainly consistent with the fungal nature of it. They thought it looked similar to something like a rhizomorph that you see with armillaria. But the other interesting thing is you have these cells. And those don't look like fungi at all. Those look like algae. Looks like something you'd see in the lichen. So maybe we'll get a better interpretation of prototaxites in the future. I think a lot of you know this story too, but I can't go by without mentioning it, which is that there is a theory, and it's pretty credible, that even the colonization of land by plants required the presence of fungi. Because when you go back to the earliest plants, earliest vascular plants, and you do a thin section of the rock, that is, you shave it so thin you can put it under a microscope, and what you see looks a lot like glomus VAM fungi in the roots. Right where you would expect a mycorrhizal associated to be. And also, you go to modern times with early vascular plants and non-vascular plants, and you find the same structures, right? 400 years later, the same two partners are still together. Here's one even older, so we're getting back there now. And I don't know, someone shows me this and says, yep, it's a fungus. I don't know, the mineralization, crystallization, how are you really going to know? Maybe it looks a little network right there. But they went an extra yard. They did X-ray absorption spectroscopy with chitin, and they actually get a little bit of a peak that correlates with chitin. Not overwhelmingly convincing, but a good suggestion that we can maybe apply molecular or physical techniques to really validate that some of these fungi are really fungi. Here we are even older, And now I have no problem. That looks like one of the blastophagiales to me. It could be an oomycete or something else, but it's certainly a fungus or a fungus-like organism. We also found cross walls, which suggests that the fungi bulbous areas, and of course these terminal ones, and in the blastula, that's where the zoospores are produced. And they did another technique, which is kind of microscopy, and they too found, where am I? Chitin sand standard. They found peaks for chitin sand, which is a deacylated chitin, and chitin. Very similar in the fossil. So another chance where now maybe in the future we get a little bit more certainty that these fossils are really how we're interpreting them. And then finally, the oldest one. This one certainly created a big stir when it was published, because it was published, this is older than most people thought the fungi were. We do have starings that go back to 2.7, but still pretty early. And, I would say controversial. But what they point out, so I've highlighted it here, is that it's not just filaments. They're filaments that are anastomose, like a fungus mycelium. They're filaments that have Y joints, they have T joints, other things that you'll find when you look at a fungal mycelium. But they don't have any chitin assay for it yet, so it's one I hope we hear more about in the future. This is also one that possibly is causing at least a misinterpretation that I've heard, which is that fungi colonized the land, like they were the colonizers of land. I think I heard that in Fantastic Fungi. I think I heard it on some Stamets YouTube video. Well, colonization requires primary producers. You can't colonize without it. Fungi, there are no known fungi that are primary producers. So they may be in very old basalts, but they can't colonize the land by themselves. What you need is a primary producer like a cyanobacteria. And in fact, there are very old sediments, similar dimensions, and similar kinds of composition that are very suggestive of microbial mass, having colonized way back then. Okay, so we're coming to the end. So what kind of conclusions can we come to now about what the most ancient one we can kind of piece together, what its properties might have been, and how they relate to animals? Well, starings was 2.7. We saw fossils going from 2.4 to 0.89. That's the fossil record. We have excellent evidence of eukaryotes with diploid and mitosis and meiosis based on the phylogenetic trees. Cytoskeleton and microtubules, flagellated single 9+2 flagellum is present, and that's the most ancient relatives of fungi. Mitochondria, yes. Anaerobic, hydrogen generating, and or oxygen respiring. That's an ongoing debate. It's pretty interesting. Chitin synthase, I know it had to be there. I just don't think we've traced the right one yet. Vesicular trafficking, I think that goes all the way back to the Milky Archaean, right? I think they have the genes for it. We hadn't recognized that until relatively recently. And we did see the amoeboid right there with the philopodia. And in some cases, these are even branched. So again, similar to a fungal mycelium. So final conclusion, most like, but not exactly, nuclearidium. And that's the end. So
I have this oversimplified version, so I wanted to know what you would think of this.
okay, well, I don't know if everyone's going to be able to see it. Sure. I mean, it's not wrong because it's got single posterior flagella. So that's kind of our definition of Opisthokonta. Has one going off to the fungi. Not a lot of details. I guess that's true. I mean, we see it, again, Linwood like that, but, because these don't have single polar flagella. But there's loss of motility. So that's okay with me. Choanoflagellates is the group of protists that's most similar to sponges, and their DNA does, trace to sponges. So they are at least the origin of animal sponges. You get the name choanoflagellate because they have a polar flagella, and then that goes off to animals. So there, I could have saved us a lot of time. Any questions?
I was also wondering if you had any hypotheses about selection factors that led to
Yeah, that's an excellent question. And, I don't think it's fine-grained enough to know all of the geology, but almost all changes have to be driven by something, right? I mean, changes in the atmosphere, changes in volcanology, sea level rise, changes in habitats. And so I don't have a good answer. The one I was looking for is the biggest one, which is the mitochondria really aerobically driven by the evolution of the cyanobacteria, and then later even accelerated. So each of those oxygen bumps almost certainly accelerates life in the aerobic form. So that's a kind of geologic correlate that has to be changing evolution. It can't not change evolution. But specifically to an Adelphida, well, there's only 10 of them, and they're all parasites of algae. So your correlation there is after the origin of algae. The microsporidians are all parasites too, and now they're parasites of things like fishes, but they're probably parasites of other things, and the host has to evolve before the parasite can evolve to whatever you want to say, parasitize or prey upon it. So the change in one phylogeny can cause a change in another. What was the other one? I didn't do the cryptomycota. I don't even know them that well. And then, of course, you could say that the origin of the glomerulomycota is the most recent one before them is the zygotes and the chytrids. Chytrids are the water molds. The zygotes came on land. They're decomposers. They're your strawberry rotting fungi. And then what's the evolutionary pressure for transition from zygomycete to glomerulomycete? It has to be the host, because they have a host that goes from non-vascular plant to vascular plant. And the hypothesis for the origin of land plants is that fungi help facilitate their ability to colonize the land because they have a superior ability to obtain water from fine locations and to obtain nutrients like phosphorus and nitrogen. And so a green alga, like I can't think of a name of a seaweed, sea lettuce. Sea lettuce isn't going to move onto land and become a plant till it has some form of root-like structure. And I think most people think that's the driving force for the evolution of the glomerulomycota. And then the basidios, well, I mean, they like polymers, right? They like starch, they like cellulose. Some of them later on evolved to degrade lignin, which is something that Bob Cummings has talked to us about before. So their substrate, if they have a substrate and a mode of life in common, that probably might be telling you that that's their evolutionary origin. But of course, you can get fooled if they maybe did that later. There's no absolutes in this kind of argument. Trying to think of a question for myself.
Oh, I'm thinking now about some of the chemical diversity that we associate with the fungi, but include they're not as chemically diverse and secondary.
There's a question. I actually, I was reading some of these papers and I had to get up and go. I'll see if I can find it. Here it is. So, one of the interesting papers I came across made a little diagram about genome size and the different groups of fungi. So, biggest range of genes, ectomycorrhizal fungi. Smallest range of genes is going to be a highly specialized fungus, like an orchid fungus. Does it have the pathogens, which is also very narrow? I know you can't see it. You have to just believe me. It's very narrow, and it's smaller. Well, pathogens don't need a lot of genes because they get everything from their host. The microsporidium is almost like a virus. It injects its nucleus, right? What does COVID do? It injects its genome, right? Its capsid fuses with ours. Its nucleic acid, which is RNA, comes in and takes over the cell. So they only have 25 kilobytes, very small. Small fraction compared to what we have. Influenza just has like 10 or 12 pieces of small RNAs. All the rest of the machinery is taken over. They take over the host. So that kind of relates to what you're saying about the chemical diversity. There are a lot of molecules that are still out there to be discovered. There are things called polyketides and non-ribosomal peptides that are really just being explored. And so it would actually be interesting to dive into a chytrid genome and find out what polyketide synthesis they do or don't have. I don't know. It has to be a question you ask first, and then you can answer it.
Do you have any examples of the chytrids we see,
Oh, so I thought perhaps I didn't go into the biggest chytrid story, which is the one that kills frogs and amphibians. So that's a worldwide disaster. You can see that here in California. It's been in California for quite a number of years. I think it's mostly like up in the Sierras. Does anyone know specifically? So this is the one they call BD, and I don't know if I can pronounce the name. It's like, what is it? Dendrobetochytridium? Something like that. And dendrobitidae. So it gets its specific name from the dendrobitid frogs, which are the poison dart frogs. That's where it was one of the first places it was discovered. And so you can go back to the 1970s and you can read our leading journals, and they're all guessing about what's killing frogs. And the biggest culprit / whipping boy was acid rain. So no idea that it was a parasite, but that's being spread worldwide. And there are multiple clades of it too.
We also have CPP and papillata, which
Is that the salamander one?
It occurs on the speed that we have here. That's the lead. It looks like the tiny cranberries on this leafy bird. But we can say you can find it in these very dry places.
Okay.
So it's not, it's a picture of the sun.
Well, maybe you could send it to Jess and put it in the work.
Oh, I had it on the table here.
Oh, you do? Really? I missed it, but maybe I didn't appreciate it too. Yes?
Fantastic presentation. I wanted to ask, first of all, what questions are you currently answering on CSUN? And secondly, when you show the hypotheses, I was wondering, do people know more about cellular differentiation fungi? For example, it depends on that 80%.
Oh, good question. I mean, I'm going to go back to my first slide. The second slide. I often ponder what they're doing, what they're quote unquote thinking, how they're responding to the environment. So I used to study bioluminescent fungi, and the bioluminescent fungi are nice as an experimental system because they give you a response that you can read very quickly, because you can photograph them in the dark, or you can put them in a photometer that actually measures light, and you can see how they respond to different things. And I've always thought that was a good system, but I stopped working on that quite a number of years ago. And I guess the question is, what chemical inputs do they respond to? They do respond to a number of different things that you don't expect fungi to respond to. So a lot of these, the reason they're making those is they're responding to light. Those are diurnal cycles. And if you put them in just dark, they're not going to make them as frequently. They may be programmed to continue to make them. I have some fungi that stop making those bands at all. In fact, some of the luminous fungi stop doing them. So they're actually sensing and responding to light as an example. And they probably respond to many other things, but to the best of my knowledge, it hasn't been elucidated. They do a lot of things. The center is different than out here. And it's like a little animal. You could call it maybe a parazoan, because it acts as a whole, right? This part knows what this part is doing in the fungal sense of knowing, which is that if there's more nutrients here and we had nutrients here, they translocate nutrients towards the growing end. The backside can actually regress and die, and they can slowly move away from that part of it. The hyphae crumble up, they reallocate as much of that as they can. So it truly is connected to each other, but how it's making that decision, other than really simple inputs like food, isn't clear. Yes?
I was going to ask exactly in that top right slide, because just a little thing you said, it's very it's a different structure than the other slides.
So this is two fungi. One's white and one's black. Yeah, that's what's going on there. And, I just let them go at it.
Yeah.
Well, you see quite a lot when you look at these contaminated plates, especially the really heavily contaminated plates. Everyone knows the Fleming story of discovery of penicillin. It was literally just penicillium fungus that landed on a plate that had Staph aureus on it. Well, if you're willing to look at the contaminated plates, that happens all the time. I see that probably at least 10 times a year looking at contaminated plates. You can find antibiotic-producing bacteria, antifungal producing bacteria, ones that they know where each other is ahead of time. They do have pheromones. Sometimes they maybe can recognize the presence of each other, sometimes not. Trying to think of a genetic system that would allow you to answer that question, Justina. But that's another thing. Fungi as genetic systems are not, there's a reason they work on yeast. Working on a mycelium is difficult in the sense that if I want to mutate a gene, you have different methods of doing it. I have to find the one mutated gene. But what if I have a string of a filament that I can't break up into individual units? I have a combination of my mutation and all the things I don't want. And it can be much harder to find certain mutations in fungi. Neurospora was used to actually do some of the first genetics. The whole idea of a gene comes from Neurospora. But that was only oxytrophy. Right, the need for certain amino acids. That's as far as they could take that. It did win a Nobel Prize. It's called the one gene, one enzyme hypothesis, back in the 1940s. Yes. Good.
Let's say science fiction, fun planet, golden rock zone, something like Venus, super high content of CO2 in the atmosphere, and let's say it's just cool enough that right there, and you wanted to see it. Let's say the planet had 50% water, oceans, 50% land. If you reach in that bag and start throwing organisms and stuff in there, and you want to get that to work properly, from the list of organisms you have there, going back to the whole list that you have currently, what kind of mixing do you have if you wanted to basically convert it over eventually to more of an oxygen-based?
Yeah, well, that's a question other people have written about and published. So one of the, it goes back to this idea of trying to change the Martian atmosphere. So Mars is very cold overall, and one of the ideas was the endolithic lichens from Antarctic dry valleys. So they actually live inside a rock. They live inside of incredibly cold temperatures. They have nitrogen fixation, and they're oxygenic. So in theory, something like that, enough of it, could eventually change something, maybe. So I don't think I have anything like that out of the fungi. Yeah, so, well, you need oxygenesis, right? And early photosynthesis was non-oxygenic. I didn't mention that, but there's, the purple sulfur, purple non-sulfur, green bacteria that are non-oxygenic, all before cyanobacteria. It was really the cyanobacteria that began the transformation to Earth's atmosphere. It's probably endosymbiosis that led to plastids that was the second one. And it's probably fungal symbioses with plants that led to the third major one. thinking too, it's like so much time. It's like we've been time traveling. Well, we can stop here. Thank you very much. Make my voice be better. I think it's what I just said.
Yeah, here's the. Yeah, I've seen it before.
Oh, thank you. Hey, good to see you. See you at school. You have the slides.
Mm-hmm.